In in 1928, Edgar Adrian put an electrode in the retina of a frog, and he knew that there was some electrical business going on there, but he heard some flicks, some clicks, and that's how he discovered that the neural system generates some electrical pulses. So, back in 1928, Titanic sank in 1920. Leonardo DiCaprio got drowned in 1920. He didn't live to see that the brain generated electric pulses. And this was used by Edward Edwards in 1973, who put electrodes in the monkey brain. The monkey was alive and behaving and performing a motor task. So he wrote impulses from the monkey brain, and this research was very interesting. I believe we need to get a sense of direction here. And this direction was given to us by Georgiopoulos and colleagues who trained monkeys to move a lever in different directions. He also observed the spiking of neurons in the monkey brain, and he observed that depending on the direction of movement of that lever, of the joystick, the neurons spiked at different rates. For example, when the monkey moved the hand to the left, the neuron spiked faster. But when the lever was moved to the right, the neuron spiked less frequently. You can also notice that the neuron worked very well when you could see desynchronization in the EEG. And Georgiopoulos, they didn't really care, and people like him don't really care about rhythms. And by the way, I have alpha rhythm, but they didn't really care about that. They only cared about the frequency of neuron spiking. And they tried to decode this. They can use this for BCI applications. But it's not enough to decode just one neuron. You really need to have information from multiple neurons. Miguel Nicolelis and John Chapin did just that. They implanted electrodes into rat brains, dozens of electrodes, and they recorded from several dozens of neurons. And they showed that this rat can control a robotic manipulator. That's how this implantation is done in monkeys. The cranium is open, you see the surface of the brain, then you put electrodes in, and then it's all glued back together with dental cement. And then you have a cap with connectors. You can connect the wires and record neural activity. That's the more recent version of this implant. This cap is 3D printed. And if things work out well, this surgery went well, the implant was not rejected, we can record from a large number of neurons at the same time. So we have about 400 neurons here at the same time. And then, using this setup, we can implement an invasive neuro-computer interface. We record brain activity, we send these signals to the decoder transforms brain signals into a signal that is understood by a mechanical arm, and then this arm can be and then we can send a feedback from this arm back to the brain. For example, trying to simulate some tactile sensation that you may feel if you had a real hand. Okay, this was a classical experiment back in 2003. Here a monkey controlled a robot using a joystick, and then an algorithm was trained, and thanks to this algorithm the monkey could control some mechanical device using its brain signals. Now a little bit of math. Here you see time on the x-axis, and you can look back 1 second. We divide this window, this signal, by 10. We compute the number of spikes in each window, then we multiply the number of spikes by the weights. We sum up all the weights, and then we use linear regression to get the signal we want. For example, we can predict the direction of hand movement. There are other algorithms as well. They are based on the same principle, in fact. I'd like to apologize for a somewhat busy slide, but here I'd like to say that if we look at t when we want to predict the signal, if we look at the time before and after the signal, then the characteristics of neural spiking, they change. There is a certain dynamics to how they spikes. Sensitivity to direction, it changes depending on the delay, depending on how we look, whether we look into the future or into the past. This is important. A lot of stuff going on here. This is the measure of change in sensitivity to direction, depending on what the subject does. Here, the monkey was controlling a robot using the hand. Then we switched on the BCI on that monkey, but then the monkey continued to use the joystick to control, or as Alexei Asachi would say, it continued to use a behavioral proxy. But then we took the joystick away from the monkey, and this monkey tried to control a robot without a behavioral proxy, directly with its brain activity. And that led to a complete change of the characteristics. We don't have any dynamics. We see many neurons were kind of indifferent to direction, became indifferent to direction. And this is very often overlooked. I very rarely see the analysis of this dynamics, and that should be done, and I hope to see this in the future. Yes, please. Nothing changes, everything is the same. By the way, I forgot to tell you, there is a problem that I will say, that I will mention later when I get to other people's studies. Otherwise, I shouldn't discuss the problems of my own studies. So, this is a very good neural interface. We record the signal of a human being. He or she holds a pen and writes something on paper. Well, things are great, but there is a but. It requires us to know well what we decode based on the brain activity. Here we decode handwriting, and we kind of push the idea, that we advance the idea that neurons encode writing. We decode this, and we have the handwriting. But in reality, things are more complicated. The brain, in fact, performs many things at the same time. For example, Julius Caesar could write, dictate, and read. Well, modern people can do that too. For example, here this lady can do many things at the same time. Or, for example, simultaneous translation that is trying to translate what I'm trying to tell you. And the same thing applies to the brain. Let me give you an example. We conducted an experiment where we separated the representation of direction and the brain representation of a motor goal. Usually, but I should say more often, these two directions they coincide. For example, if I want to take something, my direction is aimed at this direction and my motor goal is in the same direction. But things can be different in life. For example, I may be monitoring a certain signal and getting ready for a certain action. When there is a signal, I conduct an action in a completely different location. When we worked with monkeys, we put a robot in front of a monkey that commanded the attention of the monkey. This was a feeder with some tasty bananas, and there was a stationary feeder with a banana in it, but the monkey was not allowed to just take the banana. When the monkey tried to take the banana just disappeared through a hole in the bottom. The monkey was supposed to attentively watch the movements of the robot, either towards the monkey or away from the monkey. And when the robot got close enough to the monkey could either take the banana from the robot, or in the other task, it could take the banana from the stationary feeder. In that case, we saw a difference between the direction. We made sure that there was a difference between the direction of the attention and direction of the motor goal. So there was a stationary and a robot feeder. We could repeat this experiment to get the necessary statistical power. So the robot could be to the left of the monkey, less to the left, to the right, and to the extreme right. So these changes could be described by this 4x4 matrix where columns represent the position of the robot and the rows of this matrix represent the position of the stationary feeder. Now, if we look at the neural spiking, we'll see that this neuron, for example, is very active when the robot is to the right of the monkey, and not very active when it's to the left of the monkey. This neuron doesn't care. For example, this neuron doesn't care about where the stationary feeder is. This neuron, in fact, encodes the position of the robot. You can color code the cells in this matrix. The red color means a higher rate of spiking, and this strip means that this neuron really cares about the position of the robot in space. But this neuron cares more about the position of the stationary feeder because there is a horizontal, a row, and if the stationary feeder is to the left of the monkey, then this neuron is highly active. And this neuron doesn't care about where the robot is at that point of time. Now, if you put together all of these situations,
the task that the monkey had, how far it was until the monkey grasped it, we could restore the coding properties of different neurons. In this case, this neuron represents attention, because in all cases we see the vertical line, which means that the neurons were interested in the robot. In all cases, with one exception, when a robot was no longer interesting to the monkey, it was trying to grab the feed from the stationary feeder. This neuron was interested in the motor task. When the task was to grab the food from the robot, the strip is vertical. When it wanted to take food from the stationary feeder, then the strip is horizontal. We also observed neurons that were sensitive to the direction of the eyes. This neuron has the horizontal strip when the monkey is looking at the stationary feeder, and the vertical strip when the monkey looks at the robot. Now we can check how many neurons are in what parts of the brain, and it turns out, to our surprise, that the neurons that represent the motor goal are few. There are quite a few neurons representing the spatial location of attention, and many neurons that simply represent the direction of the sight. Another example, but in this case we looked at two other variables. One had to do with spatial attention versus spatial memory, and this monkey had a screen in front of it with a target that would appear in a certain position and that would rotate to a different position. The monkey needed to remember where the target appeared first, and then it had to pay attention to the final position of the target, because if that target, if it was not bright enough, it had to look at the target. If it went up, it had to remember where it appeared first. It had to look at that location where it once was. Again, 4x4 matrix and different neurons. This neuron was responsible for the monkey's attention. We see the horizontal line here. This neuron represents the memory of the monkey, horizontal line here. And this one is of a hybrid nature, because we see both vertical and horizontal strips. The neuron is part of remembering and part of paying attention, spatial attention. If we look at all the different neurons with different properties, in the dorsolateral prefrontal cortex, most neurons are responsible for attention, a few represent memory, and 24% of them have hybrid properties. And again, we separated these different variables, and we found out that it's heterogeneous. The population of neurons is heterogeneous. Some represent one type of variable, another type of variable, and those who represent both. This population can be used for decoding. I decoded the position of the target that the monkey looked at, and I also reconstructed memory, what it remembered. Also, an example with two variables. Here the monkey had a screen in front of it and was performing a simple task of moving the joystick towards the target, but the target could jump elsewhere after it first appeared. The monkey is preparing for a movement, the target appears, and then it jumps to another location. We have two targets like that, 1 and 2. Again, our 4x4 matrix, and here we see how this neuron responded to the first target first and then switched to the second one. If we look at different colors, First, you get vertical lines, and then it is transformed into a horizontal one. You would think that the monkey should forget about the false target, but if you look carefully, the monkey didn't forget. There's still a trace from the first target. Again, two variables. In this case, the monkey controls two virtual hands, either using joysticks or directly through neuro computer interface. If we look again at the 4x4 format, here the properties of neurons are quite complex. We recorded some from the motor cortex and from supplementary material, which is called in Russian additional motor cortex. Could you show us this video, please? This is how this interface works. The monkey does a good job of controlling two virtual hands. It does it quickly without suffering from any delays or low speed of information transfer. This is a direct control from the brain. Then another example of two variables. One is a motor variable. The monkey controls the virtual finger using BCI. Another one is the artificial feedback that we send to the monkey's brain. It had two gray rectangular shapes in front of it, but behind that vertical strips were hidden. The monkey didn't see it, but it could feel it using this virtual finger. Every time when this virtual finger crossed the hidden strip, the monkey got the stimulation in its sensory and motor cortex. On the left hand side is what the monkey saw. On the right hand side was the monkey had to find strips with better spatial frequencies. As you can see, the monkey scans the strips using the virtual finger, and quickly enough, using the stimulation that we send to its brain, it understands where the strips are. It uses joystick, but if we look at this other view, this is the honest brain machine brain interface. The signal goes from the brain to the virtual finger, and then when the monkey scans the rectangular shapes, it goes back to the brain in the form of electrical stimulation of sensor motor cortex. I'm going to skip that, but I have to mention that the quality of neuro-computer interface work can be assessed using these qubits of information, or something that can be calculated using the formula. I don't enjoy putting formulas into my slides, but I work with Alex Sasachi. Now I have to do it. Using these formulas, you can calculate the transfer of information in the NCI or BCI. Some make mistakes because they don't know how it works. The monkey has one target, for example, which is selected in the beginning from 8 potential targets, but it starts working, it is allowed to cross other locations. That's why it makes no sense to compute information or calculate information, but some authors still publish these results. And of course, you could think about how to record information in this experiment. You can monitor the movement of the cursor over the screen and see how close it is to different targets. And depending on that, you can guess whether the interface guessed where it needed to move or not. And you can imagine an interface when it's sure it did the right choice, it removes for the monkey a human control and does it independently. Here's another important point. A lot has been said today suggesting to use neuro-computer interface to stimulate plasticity in the brain and deal with the consequences of a stroke. These are some monkey results in terms of plasticity, but there is one problem in this case. When we switch the work of interface into the interface mode, all sensitivity of the neuron to the direction depends on the formula that we used to control this interface. Any attempt to assess the sensitivity of neurons to direction up front, straight in a straightforward way, is incorrect. You need to think twice before you could report that you calculated some plasticity coefficient or some improvement of control of this interface testifying to the brain plasticity. That's a separate issue. You could read those commentaries to get to know the arguments better. It was easy for me to make that kind of commentary by not attempting the results of Nobel Prize winners. In 2014, some scientists got a Nobel Prize for studying the sensitivity of hippocampus neurons and entorhinal brain cortex to the position of a rat in space, and they found those neuron arrays or meshes, and the rat could be anywhere in different points of that space, and it got this kind of array of neurons in the brain of the rat. There was this map of the space which it can use for its purposes. What is not clear from those studies is whether there was any control over the behavior of the rat. The rat was moving in a certain pattern way, and it was quite chaotic. You can see some nodes where the rat was more often, which looks like nodes in that neuron mesh. And together with Alexei Pimashkin and Alexei Asachi, thought about it, and fortunately we didn't have to use rats for our experiments because this data is available open source style, and we got the results of Mizuzaki laboratory. We looked at the rat's movements and how it kept moving around the arena, and one could see that the behavior of the rat is characterized by this grid-like or mesh-like pattern. We were quite happy. We uh, we it's not our specialty. We thought it would be interesting for those who study that, but you know, uh, our reviewers wanted more. They said compare it with real neuron arrays or grids that you recorded, and in this case these are the movements of rats around the arena, the example of neuron mesh, and this is the probability of a position of a rat as a function of spatial position. The neuron grid is more regular, but it still looks similar. Okay, the reviewers wanted more from us. They wanted us to give them even more. We are all physicists and mathematicians. We can analyze the divergency, and in this case we calculated the rotor from the movement of the rat. This is the grid we got. Then we calculated the divergence, and then we saw this grid looking similar to neuron discharges, and we did cross-correlational analysis and of our values for the movement of the rat and the discharges of neurons, and we saw these nice patterns that are often published in that literature. I'm not happy about these pictures because there are some hidden problems here, but anyway, we raised that issue, and also we asked ourselves what can be missed in this case. We don't deny that the brain of the rat could have some kind of map of that space, but perhaps we need to look at other aspects of its behavior. The rat, when it was moving around the arena, it marks the territory, and after it leaves its fluids there, it can modify its movement and you can monitor this movement using how it behaves. You can look at its marks and also additional behavior, sniffing, moving its whiskers and things like that. Monkeys can also navigate similarly to rats. If you turn on this video, in this case we have two monkeys. One is using a cart, another one is looking at it. Why we do that? We want to know how the brain of the monkey imagines its movement in space, and we're interested in how the brain of the observer imagines the movement in space of another monkey. And we also are interested whether you can combine several brains into one interface control, which we call brain plus brain interface. It could be two brains. It's like one head is okay, but two heads are better, or even three brains.
In this case, two brains control the one virtual hand. One brain provides one noisy signal, the second also noisy signal. When you sum the two signals together, you get a lower level of noise, you get better control of the robotic arm, of the virtual arm. We have another setup in which one monkey controls the x coordinate, the other controls the y coordinate, and they cooperate in order to control these tasks together. Or, in the case of three brains, we have primitive examples of a superbrain. Each monkey has a 2D display, but all of them taken together can control a 3D, can control a virtual hand in 3D. So you can imagine that you have a super brain. Each component of the super brain doesn't know what the other component does, but they somehow work together. Well, that's it for me. Thank you very much. That was a very optimistic note. It turns out rats can negotiate, people can negotiate. Then if we use neural interfaces, we can join all of us together in one Uber network. If somebody wants to do an experiment on yourself, welcome, come to us. There is this task called visual search. When you have a lot of things on the screen, you need to find it, to find relevant subjects. If several people do this together, they will be much more efficient in that visual search. For example, if you look for a spy with a green suitcase, one person is asked to look for a suitcase, the other person is asked to look for a green suitcase. And if several people do this at the same time, they will complete the task in a shorter amount of time. You can replicate the same model in using a neurocomputer interface. For example, we can connect our leaders and finally things will start working out well. Any questions to Mikhail Lebedev? Yes, please. Over there. Into the mic. Did I understand you right? That you were a little bit skeptical about grid cells. You said that the bright cells, the bright spots on that drawing, were where they would find their target, but the dark spots were rarely visited by the rat. Indeed, you can see this in some pictures, but in other cases things are a little bit more complicated and not exactly as you put it. Irregularity of grid cells. They look like a crystal, and it's absolutely true. If you look at the behavior of a rat, it's much more chaotic. But it has some irregularities and periodicities that emerge if you use more advanced analysis, for example, a vector field analysis. It's not about the presence of it or not presence of it. You use divergence, you use curl, and advanced mathematical tools. But what those spawns mean, they mean that the divergence is at a very low level in those places due to the distribution of paths. Well, it looks like it's not exactly a map that the map carries with itself and checks its movement against that map. But it has to do with something else. I think it's a separate story, but the rat moves not exactly chaotically. It likes to go in circles, but it prefers not to have highly curved trajectories, and that it makes a certain number of steps. But I'd like to say that you cannot extract information directly from this data. We were not able to do this. Perhaps somebody else will find, but we have not been able. Perhaps it does some snipping. Maybe there is some map, or there are some behavioral aspects of that. Off mic. Well, I think I should describe the story of how we got to this analysis. We conducted experiments on monkeys. Monkeys were driving a cart or a trolley, and we saw that some neurons in monkey cortex in M1 looked like position cells. Not exactly, but similar, and we have published a paper about this. But when we were in communication with the reviewers in fact really protected their theory that the hippocampus was the higher area of the brain collecting information from other areas in order to encode some spatial position, the sense of space, and so on. So we had to critically look at this to understand where this theory comes from. With respect to memory, it's a separate story. The Nobel Prize was awarded for the spatial map. Memory is a separate thing. So maybe we should look into it too. If we talk about humans, the hippocampus is a space it only accounts for a very small portion of the brain, and why memory is attributed to the hippocampus, it's not entirely clear. Not entirely clear. Well, according to the modern theory that you can find in handbooks, in textbooks, we get some disparate informations, and this information has to be quickly memorized. When it's not integrated, it goes into the hippocampus where it's stored for a while, and then after a certain period of time, this information is transferred to the long-term memory from the hippocampus. That's the modern view on this. You said that you talked about the prospects of synchronizing the brains of people, animals, of teamwork. Do you think it's possible to identify the Ringelmann effect, the reduction of coefficient productivity or efficiency with the growth of the number of people in this system? That's what we observed. That's what we observed when we worked with 3 monkeys. One monkey realized that it could just relax while the 2 other monkeys did all the heavy lifting. But in fact, that increases the efficiency of this collective interface. So all social tasks can be modeled in this context. Have you ever thought that there may be some external factor, for example, electromagnetic fields, the intersection of the field lines? I'd like to say that these rat data were just taken elsewhere. These are not our data, and there is the new trend that requires or that encourages scientists to share their data. That's what we used. But I read these papers carefully, and perhaps the reviewers required from the researchers to check for electromagnetic interference there were no electromagnetic influence in those experiments, and the researchers did their due diligence when it comes to measuring the magnetic fields. So there were no magnetic fields. Well, of course, these experiments were very solid, very rigorous. Well, it's possible that there were some other factors at play that may bias the results. I can tell you that when a rat goes in the maze, it is connected through a wire, the wire goes to the top, and the wire can understand where she is based on how tight the wire is. But of course, recently wireless devices have been used. Perhaps this is less relevant.